certain genes that play a role in cancer. So these three genes, when mutated, can lead to cancer. We're going to focus on proto-oncogenes first. So proto-oncogenes are normal genes that we all have, and their job is to help our cells grow by initiating cell division. If you mutate a proto-oncogene, you form an oncogene. So an oncogene is a mutated proto-oncogene. Now, oncogenes everyone do not have, because now these are mutated and these can lead to cancer. They do it by removing the regulation placed around proto-oncogenes. So now they are able to just grow at will, or not grow, but cause cell division at will. So in our oncogene pathway, so you have to have some kind of a growth factor. And this growth factor is stimulating cell growth by activating cell division. Well, it binds to its receptor, then it causes a cascade of events that leads to the activation of a transcription factor, which causes us to transcribe genes that tell the cell cycle to begin. Well, all of these have been identified to be proto-oncogenes that can mutate into oncogenes. But look, you can have mutations here in the growth factor. You can have mutations here in the receptor. You can have mutations here in the cascade intermediates. You can have mutations in the transcription factor. So all of those can be mutated to cause an oncogene, which can cause cancer. They do this by acquiring a gain-of-function mutation. Now, we've talked about those over and over again, right? Well, how exactly does a gain-of-function mutation lead to cancer? One, it can increase the amount of the encoded protein, okay? So you increase this protein. Well, if this protein is the growth receptor or the growth factor, now you have too much growth factor that's all going to bind and cause cell division. You might increase the number of the receptors. Now you have a lot of receptors to recognize the growth factors and to cause this cell division. You can have a structural change that causes something to be overly active. So say one of the receptors acquire a mutation that changes its affinity for the growth factors, makes it even more highly able to bind. Well, now you're going to have an abundance of cell division. Or you can start to express one of those in a cell that doesn't normally have it. So now you can be changing or even turning on cell division in a cell that has little to no cell division. So these gain-of-function mutations tend to be dominant, and that is something that you should automatically say, hey, you only need one copy, right? So you can get one mutation in this DNA that can cause cancer. So one mutation in an oncogene can lead to this overactive cell division. There are several different types of mutations that can occur in oncogenes. One is a missense mutation, and you should be able to define that. The second is this gene amplification. So we increase the copy number of our gene. That's going to cause us to increase the proteins or increase how many we're making. You can have a chromosomal translocation. So we'll talk about this, but like location is everything. Remember position effect? So we are going to change where our genes are located, and that can cause an abundance of activation. Or we can have viral integration. So when the virus integrates itself into the DNA, it can do so next to a proto-oncogene, putting that proto-oncogene under the promoter or enhancer of the virus genome, which can cause an overexpression. Let's look at the chromosomal translocation more. So remember we talked about CML, and this is a translocation between 9 and 21. Well, when you translocate it, you put the genes under a different promoter. So now you have this BRCA-ABL gene fusion, which is this so-called Philadelphia chromosome. And now this fusion can lead to an increase in cell division, which is making leukemic cells at this point. Okay? And so here you can see that fusion, and now it's causing different activity. So we're translocating it and like changing its regulatory proteins, which leads to a change in how we express it. And then there are certain viruses that cause cancer. So transformation is the idea of changing a normal cell into a cancer cell. And some viruses are capable of it, but most of these viruses are inefficient at it, meaning it's going to take years and years and years before they actually change a normal cell into a cancer cell. But there are some viruses who are acutely transforming viruses, or ACTs. The first one that was discovered is this RSV in chickens. But what they figured out was these viruses themselves carry oncogenes. And those oncogenes, when integrated into the genome, can lead to cancer. Okay. So now we have talked about oncogenes and how they are mutated proto-oncogenes, which can lead to this unregulated activation of cell division, which can lead to a mass of cells, which is a hallmark of a tumor that can be cancer.